This lesson recording is going to take us through the topic of enzymes, and as per the VCE study design, what we're going to focus on mainly is the role of enzymes and how they act as catalysts in speeding up biochemical pathways. We're doing a look at the mode of enzymes and how they actually work, including how we can inhibit them through non-competitive and competitive inhibition. We're also going to look at different factors that affect the rate of enzyme activity. And we're going to have a look at the role of coenzymes and cofactors and how they assist enzymes in their function. So when we talk about enzymes, we're talking about cellular metabolism, and what we mean by that is within your body you have your complete set of chemical reactions that take place to keep you alive pretty much. And the term we give to all of those chemical reactions that do keep you alive is we call it cellular metabolism. Now, in your body, bodies are very complex things, and what we see is it's not so much we have one single enzyme acting on one single substrate to produce one single product. We have pathways, whereas we have a series of enzymes that are all linked together, where you might have the first enzyme acting on a substrate, and then its product becomes the substrate of the next enzyme, its product becomes a substrate of the next enzyme, so on and so forth, forming what we call a biochemical pathway. Almost every chemical reaction in a living organism's body will be catalyzed by an enzyme. And what we mean by the term catalyzed is it's to speed something up. So nearly every chemical reaction in your body is sped up by using an enzyme, and soon we'll have a look at exactly what an enzyme does to speed up the reaction. Of all the different chemical reactions we have in our body, we can broadly place them into two categories or two types of chemical reactions. And in VCE biology, there are two main ones that we look at. So what we look at is when that chemical reaction takes place and the substrates react with one another to produce their products. Through that chemical reaction, is it requiring an input of energy, whereas the products have more energy than the reactants we started with? Or is it actually releasing energy during the chemical reaction, where the products have less energy than the reactants that we started with? And we call reactions that require energy anabolic reactions, and reactions that release energy, we call them catabolic reactions. This slide here gives a nice little overview of what those two reactions are. So up the top here we've got an example of anabolism, or an anabolic reaction. And when you're defining anabolic reactions, you can look at two things. Firstly, you're building things up. So think anabolic steroids, people take those to get bigger. So anabolic means to build. So we're taking smaller units to make a larger unit, or we're taking smaller molecules to make a larger molecule. And like I said before, all anabolic reactions, to be able to take those smaller molecules and join them together to make a larger molecule, it requires an input of energy. And as you can see here, a good example would be photosynthesis, whereas we're building a molecule of glucose and we're using carbon dioxide to do that. So we're going from a smaller molecule being carbon dioxide and some hydrogen, and we're making a larger molecule being glucose, and to do that we need an input of energy, and in this situation that input of energy comes from the sun. The opposite to that, down the bottom here, is catabolism, or a catabolic reaction. And in a catabolic reaction, we'd be taking something larger, so a larger molecule, and breaking it down into smaller molecules. And by breaking it down, we are releasing energy, so it's catabolic. A good example would be cellular respiration. So we're taking our larger molecule being glucose, and through cellular respiration we break glucose down into carbon dioxide and some other smaller molecules, and by doing that we release energy. And in this situation of cellular respiration, we use that energy to make ATP. ATP itself is a good example of both anabolism and catabolism. So if you have a molecule of adenosine triphosphate, which is an adenosine linked to 3 phosphates, when we release the last phosphate, that molecule releases energy and becomes ADP. So ATP getting converted to ADP is a catabolic reaction because it's releasing energy. To get ADP plus or inorganic phosphate back into ATP, we need to attach the last phosphate, so we'd be making the molecule larger. And to attach that last phosphate, we need an input of energy. So going from ADP back to ATP is actually an anabolic reaction. So that cycle is a really good example of using both. Some other examples, if you're looking at anabolism, you could think of making a polypeptide chain. So going from single amino acids, joining them together through condensation reactions, forming peptide bonds, makes a polypeptide chain. So from amino acids to polypeptide chain would be anabolic. Conversely, if we go from a polypeptide chain and break that down in our digestive system back into individual amino acids, then that would be catabolism, because we're going from something larger to something smaller. Here's a table you can fill out to just consolidate what I was just talking about. So maybe at this stage, pause the presentation and try and answer those individual boxes. So let's go through the answers now. So catabolic, also called an exergonic reaction. Anabolic, also called an endergonic reaction. So exergonic means releases energy, endergonic means it requires energy. Catabolic, is it energy requiring or releasing? Well, we know it's energy releasing. Anabolic, we know it's energy requiring. Build or break down molecules. Catabolic breaks down molecules. Anabolic builds larger molecules. Biological example of catabolic, cellular respiration, among other examples. Biological example of anabolic would be photosynthesis, and once again, among some other examples. So let's move into why we need enzymes and then into what they actually do. The simple answer is we need them to stay alive. All of the chemical reactions in our body, look, theoretically, they would take place without enzymes, but they would require so much heat and so much time to take place that we'd be dead before we got the products from them. So there's a lot of ways to speed up chemical reactions, and heat is one of them. But our body needs to stay at 37 degrees Celsius. And at 37 degrees Celsius, the chemical reactions that we need to take place immediately in our body, they're going to take days, and your body doesn't have days to wait, so it would not be able to function. So because we can't use heat, what we use is enzymes. So an enzyme, as I said before, is an organic catalyst, or you could call it a biological catalyst, which means it's an organic product that is able to speed up a chemical reaction. And a classic question you might get in a SAC or exam would be, could be multiple choice or short answer, how do enzymes speed up chemical reactions, or how do enzymes catalyze chemical reactions? And your answer would be that they lower the energy levels required to activate the chemical reaction, or they lower the activation energy of the chemical reaction. And what we'll look at on our next slide is exactly what we mean by that statement. So here's a graph for you to look at, which could look a little bit technical at first, but if we break it down, you should get the hang of it. So over here on this axis, we've got our energy, obviously increasing from 0 units of energy at the bottom up to the higher level of energy at the top. And along our bottom axis here, we've got the progress of the chemical reaction. We've also got shown to the left of our graph our reactants, and to the right of our graph we've got our products. So the graph is showing a chemical reaction taking place. On the furthest left-hand side of the graph, we start with our things that are going to react, and on our furthest right-hand side of the graph, we end up with our products following the chemical reaction. What you can see is the reactants have a certain level of energy to start with, and that's because we store energy within the chemical bonds of any molecule. Now, the level of energy they have is not enough energy to make them react with one another. And if they're at that energy state, well, they're just going to exist as they are in their reactant form. There's not going to be a chemical reaction. What we need to do is we need to get them to a more excited state, so get them to a higher energy level where they actually get to the point where they will react with one another. And once we get to the energy level where they become reactive with one another, then our chemical reaction will take place. So if we look at this line up the back here first, which says without enzyme, so we can see here, as the chemical reaction is going to start, we go from the starting energy level. Let's just imagine this is 1 unit of energy, and then 2, the next line, 3, 4, 5, 6, would love a stylus and a tablet screen. So what we need to do is we need to get the reactants from the energy state of 1 all the way up to the energy state of, let's call that 6, up the very top. And as soon as they get to the energy state of 6, the chemical reaction will take place. So we would call that up here the activation energy. Now, once it's reached this level of activation energy, that means that's the level of energy required to initiate a chemical reaction. Chemical reaction takes place, and then the energy level drops back down to an energy level that represents the energy left in the products. Now, in this example, the products on the graph have a lower amount of energy than the reactants. So we would imply that this is a catabolic reaction because the products have less energy than the reactants we started with. So some energy has been lost from that system, from that chemical reaction, and that's what happens in a catabolic reaction. If you look here now at the line that says with enzyme, you can see that there's a slight increase from the starting energy of the reactants at 1, but it's really only a really slight incline up to that middle point there where we reach the activation energy, the amount of energy needed for the chemical reaction to take place. Chemical reaction takes place, and then the energy levels drop back down to where they would be representative of the products. So both of these lines are using the same reactant and producing the same product. So they both start with the reactants having the same amount of energy. They both finish with the products having the same amount of energy, but by using the enzyme, the amount of energy needed to start the chemical reaction, the activation energy, it's a lot lower. And because it's a lot lower, the chemical reaction will take place quicker because the reactants will reach that activation energy level faster. So therefore the chemical reaction will take place faster. So what exactly is an enzyme? So an enzyme is a protein. Specifically, it's a globular protein. Now, if you think back to when we learn about proteins, we have our primary, secondary, tertiary, quaternary structure. So enzymes can be either tertiary or they could be quaternary, and it just depends on the type of enzyme. If an enzyme is tertiary, which means it's just one single polypeptide chain folded into a 3-dimensional shape, we would call it a monomeric enzyme. Mono meaning one, and that's relating to one polypeptide chain. And plenty of enzymes are like that, though we do have some enzymes that are not functional until they consist of two or more polypeptide chains, so until they're in their quaternary structure, and those enzymes are referred to as oligomers. We also have, so most enzymes, not most, but a lot of them are just protein. They are literally just their polypeptide chain, or their two or more polypeptide chains folded into their three-dimensional structure, representative of that functional enzyme. But we do have some enzymes that won't just work being just that pure protein structure, and they have some other things associated with them that enable them to work. And we refer to those as cofactors or coenzymes. So some enzymes require cofactors to be attached to the enzyme, and these are inorganic things such as metallic ions, whereas other enzymes require coenzymes, which are sort of like these little transfer molecules that can transfer functional groups to that enzyme catalyzed reaction, and they need those coenzymes for that chemical reaction to take place. Down here we've got a couple of models of an enzyme. So the model here marked A is showing that classic tertiary structure. You can see it has some alpha helices, has some beta pleated sheets as well, and it's folded up into a three-dimensional structure which gives it a specific active site here, and the active site is where our reactants or our substrate binds to when a chemical reaction takes place. B shows you another representation, which is showing you really the amino acids now and how they're all folded up into that three-dimensional shape. Once again, you can see that little cutout, which we refer to as the active site. And then diagram C is the same enzyme, but it's now showing you how the substrate attaches to the active site of the enzyme. And in a couple of slides time, we'll talk about some models that explain the relationship between the enzyme's active site and the substrate, and talk about how they're complementary in shape, and that relates to the specificity of the enzyme. So you've probably picked up by now that enzymes are very specific, and we can pretty much get away with saying each enzyme acts on a specific substrate. And the reason it acts on a specific substrate comes back to the fact that the substrate binds to the active site of the enzyme to form an enzyme substrate complex, and the substrate will only bind to an active site that is complementary in shape. So therefore we say enzymes are highly specific. Now, it is a bit of a stretch to say that every enzyme only specifically acts on one particular substrate, because though that can be true, it's not always the case, yet they are still highly specific. So we have three types of specificity. We have absolute or substrate specificity, and this will be the one that you're familiar with, because this is the one related to the enzymes that do only act on one specific substrate. And most of the time that's probably going to be the example you get and the way you'll talk about it. So absolute specificity or substrate specificity, that is when an enzyme only acts on one particular substrate. We do have some other types though. We have bond specificity, and in this situation it's not just the case that the enzyme only acts on one particular substrate. It's that enzyme only acts on one particular type of chemical bond. So if you take proteins, for example, we can have a number of different types of proteins, but the thing those proteins have in common is the peptide bonds between their amino acids. So peptidase is an enzyme that acts on the chemical bonds that are called peptide bonds. So it could catalyze and act on multiple protein substrates, because what it's looking for is the specific type of chemical bond. We also have group specificity, and this is an enzyme that will only act on molecules that have a particular functional group, not functional group, functional group. They'll only act on molecules that have a particular functional group surrounding their chemical bond. So, for example, if you think of a peptide bond, you could have a group-specific enzyme that only acts on chemical bonds that are surrounded by basic side chains. So basic your amino acids that have the basic R groups instead of the acidic R groups. Some enzymes will only act on peptide bonds that are surrounded by the basic side chains. Where are we going to find enzymes in your body? We're going to find them everywhere. Most of them you're going to find actually inside body cells. So most of our enzymes are intracellular, which means they live within specific body cells. So a good example could be your enzymes that are responsible for producing ATP, your enzymes that are responsible for driving photosynthesis or cellular respiration. They're all found within a cell. Though we do have some enzymes that live in your extracellular environment, so outside of your body cells. And some good examples of those would be lysozymes, which are found in your tears, amylase, which is in your saliva, thrombin, which is found in your blood, and then a lot of your digestive enzymes that are found in your stomach. Those enzymes aren't literally within a cell, they're in an environment outside of the cell. We also have this situation, obviously, because enzymes are these catalysts that can drive chemical reactions, that can break things down and build things up, we need to regulate their action pretty tightly. So many of our enzymes actually exist for a lot of the time as proenzymes, which means they're in their inactive form. So if an enzyme is referred to as a proenzyme, it can move around the cell or move around the body, and it's not catalyzing any chemical reactions, because what we need to do is we need to activate it. And we have different signals in our body that can be sent to proenzymes and cause them to become activated at the time when we need them. Here's a good example of how we can activate a proenzyme. So this example is going from looking at an enzyme called pepsin, and on the left here it's in its proenzyme form, which is called pepsinogen, and on the right over here it is in its active form, its enzyme form, in which we would refer to it as pepsin. So in its inactive form, its proenzyme form, what we've got, if you look up in this region here, there's a short blue sequence, a short blue polypeptide sequence, which is be about 40 odd amino acids long. And what we say it's doing is it's actually masking or covering up the active site of this enzyme. And as long as that masking section is there, nothing can attach to the active site of the pepsinogen enzyme, so it's inactive. Now, pepsinogen is an enzyme that is released by your stomach cells into your stomach, and your stomach is a really acidic environment. So once the pepsinogen is inside your stomach and it's exposed to that lower pH level, that more acidic environment, the change in pH causes the masking sequence, the blue region, to be removed from the active site. And once it's removed, the active site is now free to bind with the particular substrate. So we say that enzyme is now active. Another example of a proenzyme getting converted to an enzyme and why that's beneficial is in the process of forming a blood clot. So when you cut yourself, you would know you start bleeding, but pretty soon after that you would form a scab on your skin. And that occurs because your blood starts to clot. What makes your blood clot is a protein in your blood. called fibrinogen or fibrin. So when your blood's not clotting, you've got a protein present called fibrinogen, and it's a soluble protein. However, once you cut yourself, that protein called fibrinogen gets converted into an insoluble protein called fibrin, and because it's insoluble, it causes your blood to coagulate, and that's what forms the clot. Obviously, you would not want that insoluble protein going through your body all of the time, or you would be getting blood clots when you don't want them. So, because that's a chemical reaction of fibrinogen to fibrin, it is catalyzed by an enzyme. So the way we turn that reaction on or off comes back to when we turn the enzyme catalyzing it on or off. So you've got an enzyme in your body called prothrombin, and when it's inactive, it's not catalyzing that chemical reaction to convert fibrinogen into fibrin. However, when you do cut yourself, some different chemical signals get sent around your body, and they cause that inactive prothrombin to become activated into the enzyme called thrombin, and then that active enzyme thrombin catalyzes the chemical reaction of fibrinogen into fibrin, and that will cause your blood to clot, and that will stop you bleeding to death, which is a good thing. Now, don't be overwhelmed by this example. I wouldn't expect you to be able to rattle it off word for word, but it is just a good example of why we need proenzymes and why we need a way to activate them into enzymes. So let's have a look at how the enzyme actually works. So what we know is that enzymes are biological catalysts. They speed up chemical reactions. How do they speed up the chemical reactions? They lower the activation energy. Why are they able to lower the activation energy? Well, the answer is because they can take the reactants, which we call the substrates, and they can hold them closely in the active site of the enzyme in this perfect little environment that enables it to have less energy needed for them to react with one another. So when we're talking about enzymes, we've got some keywords that we must use. So the compound being acted on by the enzyme is called the substrate. The thing that the enzyme produces at the end of the chemical reaction is called the products. So a substrate comes into the active site of the enzyme, and then a product is released out of the active site of the enzyme. And then, of course, the enzymes work by attaching to the active site itself. So what is an active site? It is the specific region on an enzyme that a substrate binds to. So the specific region of the enzyme that the substrate binds to. And as you can see here, the substrate is held in place by weak bonds, being hydrogen or ionic bonds, which means the substrate can easily attach, but the product can easily be released as well. The active site, you could also refer to that as the substrate binding site, but you are better off calling it the active site. So substrate binds to active site of the enzyme, products are released from the active site of the enzyme, and we also know that the substrate and the enzyme's active site are highly specific to one another, complementary in shape, and we have two models that talks about that complementarity between the substrate and the active site. So we have the lock and key model, and then we have the induced fit model. So these are definitely two models you would need to be familiar with, and you could definitely expect a question on a SAC or an exam that gets you to compare and contrast between the two models. So the difference is our first model, our lock and key model, this was the first model proposed to explain this relationship between enzyme, active site and substrate. And much like a lock and key, it pretty much says the active site of the enzyme is completely complementary to the shape of the substrate. And that is why that enzyme acts on that substrate. because they're a perfect match for one another, a perfect complementary match. Then we have our induced fit model, which isn't completely different, just a little bit different. So it was a later model, and both models are still sort of acceptable. And the induced fit model goes on the premise that yes, the active site and the substrate are definitely complementary in shape, but there's a bit more flexibility to it than being absolutely perfectly complementary. It sort of says the active site might vary slightly to the shape of the substrate, and then after contact with the substrate induces a complementary shape around it by the enzyme. So to recap that one, you've got your substrate and your active site, which vary slightly in shape, but they're still very similar. After contact with the active site, the enzyme then fits tightly around the substrate, creating a really nice snug fit. So I know in class I always use the example of your bum sitting into the bean bag, and the bean bag being like the active site of the enzyme. That only becomes perfectly complementary after contact has occurred. Nice easy diagram here, just consolidating pretty much what you should already know by now. So you've got your enzyme, you've got your active site in there, and then you've got your substrate. And then we move down to this diagram here, which is showing the substrate bonded to the active site of the enzyme. And this is a key word down here, enzyme substrate complex. So a substrate attaches to the active site of an enzyme to form an enzyme substrate complex. Then chemical reaction takes place here, and what we're left with is an enzyme product complex, whereas you've got the products and they're yet to be released from the enzyme. And then finally over in this diagram here, you've got your enzyme, which looks exactly the way it did when the chemical reaction started, because they're not used up or damaged or modified or changed at all during the chemical reaction. They stay the same. It'll be able to go off and act on a brand new substrate, and then our two products have been released. So make sure you're familiar with the word enzyme substrate complex. That's the most important thing to take from that slide. Another example here, whereas the reaction on the last slide showed a catabolic reaction because it was taking a single substrate and the products it produced were two smaller products. This is an anabolic reaction, so you've got your one substrate here, A, one substrate here, B, enzyme here, substrates bond with the active site of the enzyme. What would you call this here? An enzyme substrate complex, I hope you said. And though this doesn't show an image of an enzyme product complex, not all diagrams will, it does show you that you've got your product released, which is a larger product than the two smaller substrates we started with, so it's anabolic. And once again, you've got your enzyme shown down the bottom here, unchanged, exactly the same as it was before the reaction started. So the ability of an enzyme to work can vary. Sometimes an enzyme catalyzed reaction will be occurring slowly, and then other times it'll be occurring really fast, which is at its optimum. Now, enzymes are pretty fussy little molecules, and they like their conditions to be just perfect if they're going to work at their best. And if we go outside of just perfect, they can either slow down in their activity or they might stop their activity completely. So we say we've got different factors that affect enzyme activity, and these factors are pH, temperature, the concentration of the enzyme, and concentration of the substrate. In terms of pH, every enzyme has its optimal pH level in which it works best. and outside of that particular pH level, it's going to work more slowly, or it's going to denature and not work at all. So for this particular enzyme, its optimal pH is indicated here because we can see the rate of activity is the highest at this point. So when the rate of activity is the highest, that's indicative of the optimal pH, or the optimal whatever factor we're looking at. Got a pH scale down the bottom here. Our pH is going to go from 1 to 14, and this picture pretty much looks like it's in the middle, so we could say this enzyme appears to have an optimum pH of 7, because at pH of 7 the rate of reaction, or the rate of enzyme activity, is the highest. If it's at a pH of 6 over here, it's still working, it's just not working as well. If it's at a pH of 8, same again, it's still working, just not working very well. However, if it's down at a pH of 1 or up at a pH near closer to 14, there's no rate of enzyme activity, which would imply that that enzyme's so far outside its optimal pH range that it's denatured. So the optimal pH, there's no one size fits all. For humans, it's typically somewhere around 7.6, but it really just depends on the organism or the environment. So our enzymes that work in our stomach, for example, their optimum pH definitely wouldn't be 7. Their optimum pH would be down in the acidic scale of the pH range. So it really comes down to the environment where that particular enzyme is found. If we're looking at temperature as a factor that affects enzyme activity, what we can see that with increased temperature, we do get an increase in enzyme activity to a point. So if we look here at a human enzyme, the rate of reaction is increasing as the temperature increases up to about 45 degrees Celsius or 40 degrees Celsius. But after that time, it drops back down rapidly to no rate of reaction or no enzyme activity. So we say enzyme activity increases with an increase in temperature until the optimum temperature has been reached. And once again, optimum temperature is indicated by the point in the graph where the rate of reaction is the highest. Over here we've got an example of an enzyme from a bacterium found in hot springs. Now, because it's found in hot springs, that indicates it lives naturally in a really hot environment. So whereas a human optimum temperature is going to be somewhere around 37, because that's the temperature of our body, this enzyme that spends its time living in a really hot environment, you can see, as indicated by the graph, its highest rate of activity correlates with a temperature of about 70 degrees Celsius. So once again, it's environment specific. So for this bacteria, 70 degrees Celsius is the optimum temperature for enzyme activity. Temperatures below 70, enzyme activity is slower, but enzyme activity increases up until we get to that optimal temperature range. And then once we go too far past that optimal temperature range, enzyme activity drops off pretty suddenly back down to 0, because what's happening is once we go past the optimal temperature range, pretty quickly the enzyme's going to become denatured. And to become denatured means the enzyme is going to have a change in its shape. It's going to be unfolding. And as soon as it starts unfolding, that will change the shape of the active site. And even if the active site changes shape slightly, it's going to prevent the substrate from being able to attach. So we would say that enzyme's no longer working, and we call it denatured. At lower temperature, enzymes don't denature, they just work more slowly. At higher temperatures, they denature. So when we look at the effect of enzyme concentration on the rate of reaction, what we see is a linear relationship, whereas theoretically, as long as the enzyme concentration increases, the rate of reaction will increase. Though this graph is a little bit misleading, because it won't happen forever, as I'll sort of explain now. So with enzymes and substrates, it's all about collisions. It's about the enzyme's active site coming into contact with the substrate. So if you've got a closed system of, say, 5 substrates and 2 enzymes, there's going to be less collisions between the substrate and the enzymes in that situation than if you've got a system where you've got 10 enzymes or 20 enzymes. So the more enzymes we add, the faster the rate of reaction, up until a point. So this graph could plateau for 2 reasons. You've gotten to a point where you've broken down all of the substrate. So no matter how many enzymes you add, there's no substrate left to act on, so the reaction's going to stop. Or you've gotten to a point where the concentration of the enzyme is equal to the concentration of the substrate. And if you add more enzymes, there's still no more substrate for it to act on, so it's not going to happen any faster. So we see a plateau. The effect of substrate concentration on enzyme activity is really similar to the effect of enzyme concentration on enzyme activity. So the rate of chemical reaction will increase as we have an increase in substrate concentration. So in a chemical reaction, if we add more substrate, that will make the reaction take place faster, once again up until a point. And you can see that we've got a plateau at this point here, and past that point, no matter how much substrate we add, the rate of that chemical reaction will not increase any further. And the reason that's occurring is because we've got a set number of enzymes within that chemical reaction. And at this point marked by the X, we're pretty much saying, well, all of the enzyme's active sites are completely occupied by the amount of substrate that's present at this level. And even if we add more substrate, there's no free active sites of the enzymes for the substrate to bind to, so it's not going to make it take place any faster. If we added in more enzyme, it would take place faster, but as long as that enzyme, all the enzymes are fully used up, then substrate concentration is going to plateau. So in terms of our enzymes, we've looked at how they work, we've looked at factors affecting their rate of reaction. Well, another thing that can affect enzyme activity is whether or not the enzyme is being inhibited or not. So there are certain things that inhibit the function of our enzymes and stop them from working. And they can stop them from working temporarily, or they might stop them from working permanently. So we call it inhibition, enzyme inhibition. And our inhibition, where it just inhibits it for a little while, we would call reversible, because it can go back to normal. And then we can also have inhibition that we call irreversible because it's permanent. And we also have competitive inhibition, and then we have something we call non-competitive inhibition. So let's start here with a reversible example of competitive inhibition. So what happens with competitive inhibition is we have a molecule that is referred to, or not that one, that's our substrate, this one down here, a molecule that's referred to as our competitive inhibitor. And what your inhibitor does is, like a substrate, it is also complementary in shape to the active site of an enzyme, and it will bind to the active site of an enzyme. And as long as an inhibitor is bound to the active site of the enzyme, you can see pretty clearly there's nowhere for the substrate to attach. So we say that enzyme is being inhibited from working because it can't attach to the substrate. So the inhibitor and this substrate are competing for the active site of the enzyme. And if the inhibitor is attached to the active site of the enzyme, it prevents the substrate from being able to attach, and therefore a chemical reaction cannot take place. The inhibitor will eventually move on and the enzyme will start working again. That inhibitor is non-covalently bonded to the active site, so it's still a weak bond, so it can be broken and it will eventually move. Now, with non-competitive inhibition, can still be reversible like competitive, but in this situation, it's not the case where the substrate and the inhibitor are competing for the active site. The inhibitor actually is not attaching to the active site at all. So on an enzyme, you can have a site that's not the active site, and we call it the allosteric site, which is this site over here. And the allosteric site is a site other than the active site on an enzyme where an inhibitor attaches to. And what happens is the inhibitor attaches to the allosteric site, and by attaching, that causes a change of shape to the active site. And as soon as the active site changes shape slightly, it's no longer completely complementary to the substrate, and the substrate cannot attach. So it's not competitive because they're not competing, but it's still preventing the substrate from attaching. So we would say non-competitive inhibitor inhibits the function of an enzyme by binding to the allosteric site and causing a change in shape of the enzyme's active site, therefore preventing the substrate from attaching and the chemical reaction from taking place. This slide's just an accidental double up from the first slide I talked about on competitive inhibition, but the question down the bottom is not a bad one to at least look at. How could you increase the rate of activity in such a situation where competitive inhibition has taken place? So remember I said the substrate and the inhibitor compete for the active site. So if we increased the substrate concentration to a point where we've got a lot more substrate than inhibitor, if they're both competing for the active site of enzymes, well, the more substrate there is, the greater the chance and frequency that the substrate will win the competition and attach to the enzyme. So you would be able to increase the rate of reaction by increasing the substrate concentration. We also have this thing called end product inhibition, and this is to do with non-competitive inhibition, and it's sort of like showing how we can use non-competitive inhibition in a good way in our body to try and regulate the production of a particular product. So our bodies, in terms of homeostasis, they like things to be just right. So we do need ways of regulating things. It's not the case that inhibition is always bad. Sometimes inhibition is necessary. So with end product inhibition, basically we're using the end product of a biochemical pathway to inhibit the starting enzyme of that pathway. So if you look at this picture, we've got our substrate. It binds to the active site of enzyme 1, forms an enzyme substrate complex, and it produces a product that we call our first intermediate substrate. We're calling it an intermediate substrate instead of a product because it's going to get acted on because it's the substrate for enzyme 2. Enzyme 2 acts on the product from enzyme 1, so the first intermediate substrate, and by doing that it produces the product we call the second intermediate substrate. So essentially the product of enzyme 2 gets acted on by enzyme 3, and enzyme 3 produces our end product. So the end product is the thing we were after in the first place, but we needed this series of chemical reactions to take place to get to that point there. However, the end product also can act as a, trying to draw an arrow here, a non-competitive inhibitor to enzyme 1. So enzyme 1 has an allosteric site, and the end product is able to attach to the allosteric site. And as we know, if that inhibitor attaches to the allosteric site, changes the shape of the active site, and therefore the substrate can no longer attach. If we've inhibited enzyme 1, well, it can't act on its substrate and it can't produce the first intermediate substrate here. And then we've got nothing for enzyme 2 to act on, so it can't produce the second intermediate substrate. And then we've got nothing for enzyme 3 to act on, so it cannot produce the end product. So I suppose think about it like this. Too much of something is not always a good thing. And we don't want to produce too much of the end product. So as we get large quantities of end product, if they start attaching to enzyme 1, that's going to prevent us from making more end product. And then the levels of end product will decrease a little bit, or essentially they'll just stay at a pretty stable level. So we've talked about reversible inhibition. With irreversible enzyme inhibition, this is obviously bad because the enzymes will never work again. You've got an inhibitor molecule that is covalently bonding to a part of the enzyme, and by doing that it's causing a permanent change in the structure of the enzyme, which is permanently affecting a substrate from attaching to the active site. So because that happens, the enzyme is rendered useless and it will no longer work. So some good examples of things that would cause irreversible enzyme inhibition are typically things like your poisons, your heavy metals, nerve gases, and like a little sign over here, our cyanide is a good example. One final way that we could inhibit enzyme activity is we could induce that inhibition ourselves. So purposely add something into an organism or an organism cells to cause a particular enzyme to stop working. And what we do is we call that rational drug design. So there are obviously different infective agents, whether they be bacteria or viruses, that can infect a person and cause that person to have an illness. Now, if those bacteria or viruses use enzymes as part of their infective process, if we can work out what enzyme they're using to help them carry out infecting a person's cell or an organism's cell, we can design a drug that can act as an inhibitor that can inhibit that particular enzyme and therefore prevent that infective agent from being able to infect an organism. So in terms of rational drug design, a type of question you would get on it would probably be a definition-based question. And this definition here in dot point 1 is a pretty good one. So rational drug design involves finding out how, and up here we should probably write enzymes, if I can. You get the point. Finding out how enzymes in an infective agent work against a cell and using that information to design a drug that prevents the infective agent from being able to do what it does. So we could use rational drugs to prevent diseases that have enzymes as part of their infective process. And essentially what we're doing is we're artificially making an enzyme inhibitor. So here's a good case study of how we have used rational drug design already. So you've got influenza, which is the flu, not really your common cold, but more the actual influenza virus, the thing that makes you pretty sick. And the flu or influenza is caused by influenza virus. And this is a diagram of influenza virus over here. Viruses are pretty simple things, as you'll see when we do the immune system, but they're essentially just a coat of protein, and inside that coat of protein is some genetic material. And what a virus does is it will attach to the outside of a cell. It will either enter the cell itself or release its genetic material into the cell. Using the cell's organelles and machinery, it will make multiple clones of itself, and then those clones will burst out of the cell, killing the cell, and then travel through the organism's body to infect more cells and do it all again. Now, what scientists worked out was that on the surface of a virus, there's a particular enzyme, and that enzyme is called neuraminidase. And what that enzyme does is it actually is the thing that enables the viruses to burst out of a body cell to go and travel further through the body. So theoretically, if a virus did not have neuraminidase, if influenza virus did not have neuraminidase, it would not be able to escape the cell it has infected. So that would help prevent the spread of it throughout the body. So what scientists have done is they have designed a drug called Relenza, and Relenza acts as a competitive inhibitor to neuraminidase. So Relenza will travel through the body and it will attach to the active site of the neuraminidase enzymes on the outside of the virus, therefore preventing the virus from being able to escape the cell. So this is an example of rational drug design because scientists discovered that there is an enzyme as part of the infective process of a particular virus, and then they've just used that understanding to design a drug that inhibits the action of that enzyme, therefore preventing it from carrying out what it was intending to carry out. So our last slide to look at in terms of enzymes comes back to something we talked about a bit earlier, and that's the idea of having these cofactors. So some enzymes, as I said before, work just by themselves as straight protein enzymes, but there's others that require cofactors or coenzymes to be associated with them to make them actually work. So our cofactors are our inorganic cofactors, such as our metal ions that attach to our enzymes. And then we also have these things called coenzymes, which is a organic molecule that has a role in transferring what we would call functional groups to an enzyme catalyzed reaction. And our best examples of coenzymes are the coenzymes NAD, used in cellular respiration, the coenzyme NADP+, used in photosynthesis, or the coenzyme ATP, which is used in pretty much everything. NAD is a coenzyme that transfers hydrogen ions and electrons to the electron transport chain in cellular respiration, whereas NADP+ is a coenzyme that transfers hydrogen ions and electrons to the Calvin cycle during photosynthesis. And ATP is a energy-rich molecule that transfers chemical energy to a range of enzyme catalyzed reactions throughout the body.